Welcome back to the series of the topics where we are discussing about the vectors. Now we will see about the Phagemid vector. So what are these Phagemid vectors? So let's dive into this topic about the Phagemid vectors. So these Phagemid vectors, they are actually the DNA-based cloning vectors. So these Phagemid vectors, they have the plasmid DNA. So they have actually the plasmid DNA, and they contain also the small segment of genome of a filamentous phage. So they have the genome of the filamentous phage. So these Phagemid vectors, so this comprises of the Phagemid vectors. Thus we can say that these Phagemid vectors, they are the plasmid DNA that contain a small segment of genome of a filamentous phage. Now these phages, the filamentous phage, this filamentous phage can be the M13 phage, or it can be the F1 phage, or it can be the FD phage. So this comprises the Phagemid vector. Now let's see fully about these Phagemid vectors. So these are the filamentous phage-derived vectors that contain the replication origin of a plasmid. And their properties include the small size, and they have the high genetic stability. They can be used to clone single and double-stranded DNA. Now looking into the properties of these Phagemid vectors. These Phagemid vectors, they contain the basic components, like it needs the replication origin of plasmid, then it needs the selectable marker, and it needs the intergenic region, that is the IG region. And this IG region, it usually contains the packaging sequence, and it needs the code protein sequence so that it can code for the phage code proteins, and it needs the restriction enzymes recognition sites so that restriction enzymes can cut out that site. It needs the molecular tag, it needs the signal peptide DNA sequence so that it can code for the signal peptide, and it needs the promoter sequence. So these are the basic components of the Phagemid vector. So this Phagemid vector, the important thing is that it needs the restriction sites. The insert is about 10 to 20 kbs of size, and important thing is that it needs the replication origin of plasmid. It needs the replication origin for the of the plasmid DNA, so that the double-stranded DNA can be replicated, and also it needs the origin of the FN phage, that is for the packaging in phage particles, so that we can package it into phage particles. And this Phagemid vector, this is actually used in the phage display technique. So this technique is the technique for study of protein peptide and protein DNA interactions. Thus we can say that the phage display technique is an important technique for protein interactions, and this Phagemid vector is used in technique, that is the phage display technique. And thus we can also summarize that this Phagemid vector, it needs the replication origin of the plasmid, a replication from the F1, so that it can be packaged in the viral particles, and for facilitating the subsequent operations, for example the gene manipulation and the protein purification. So these vectors are used. So this is all about the Phagemid vectors.